So let's just review what the large intestine does. So remember that in the small intestine is where we finished all of our chemical digestion, and it's where we did most of the absorption in the small intestine. So the small intestine is where we're absorbing all of the monosaccharides, all of the fatty acids and glycerols, all of the amino acids, and then most of our minerals, ions, most of our vitamins, water soluble and fat soluble, and most of our water. So the large intestine, this is where whatever water, minerals, ions, and vitamins that we didn't absorb in the small intestine, we can do those here in the large intestine. And then whatever remains, we're going to take that semi-solid fecal material, store it, and then eliminate it here in the large intestine. So when we look at the large intestine in terms of adaptations, there's going to be a few different adaptations that we'll talk about here in a second. And then there's some unique features that we see in the large intestine that we don't see anywhere else. So a couple of these. One is called tunics, and this is where we have 3 bands. They're relatively small bands, but 3 bands of longitudinal smooth muscle cells in muscularis externa. So we said that in the esophagus we have 2 layers of smooth muscle cells down in the lower part of the esophagus. In the stomach we have 3 different layers of smooth muscle cells. In the small intestine we had 2, and here in the large intestine we're going to have 3 layers. Now again, they're really small layers, and they're all longitudinal, so they're all running lengthwise. We also have, because these muscles, these smooth muscles, are always slightly contracted, it's going to set up haustras. And these are these pocket-like sacs that you see all along the large intestine. And it's because these guys are always slightly contracted. So instead of the large intestine just being a tube, it kind of puckers all along the length of it. And those puckerings, or those little pockets, are called the haustra. And again, it's due to that smooth muscle contracting in the background. And then if you look from the exterior part of the large intestine, you have these appendages that are hanging off. And these appendages, they don't really know what they are. They appear to just be fat-filled pouches of the visceral peritoneum, or the connective tissue that's on the outer part of the large intestine. Just kind of hangs off. And it just may be extra places where you feel, or extra places where you find adipose tissue or extra energy reserves. Why they're specifically hanging off of the large intestine is not known, and not all mammals have these, so they don't really have a good understanding of what that is. Now, when we look at the large intestine, we said it's divided into different parts. That would be the cecum, and then the appendix hangs off of the cecum. And the cecum is also where the last part of the small intestine comes into and meets up with the large intestine. So at the cecum, you'll have the ileocecal valve. Again, the appendix hangs off of the cecum, and then you go into the colon. And for the colon, you have the ascending colon. You have this curve right here called a flexure. This would be the hepatic flexure because the liver sits up here, or it's also referred to as the right colic flexure. Then you have the transverse colon, and then another curve here, the splenic flexure, or the left colic flexure, descending colon, and then on the back side you'll see the sigmoid colon kind of curves around, goes into the rectum, and then the anus down here. Now, the cecum. Again, this is where you're going to find the appendix hanging off of it. So this would be the appendix right here. And the actual function of the appendix is really not known. There's a lot of different thoughts about what's going on with the appendix. And it appears that the appendix has a fair amount of lymphoid tissue in it. So it's thought that it may help in terms of immunity and making sure that the bacteria that's in the large intestine doesn't overwhelm the large intestine. So in the appendix, you're going to find this lymphoid tissue. You also can find some bacteria in the appendix from the large intestine. And again, they don't really know what the job is. The thought that it probably plays a role overall in body immunity, but particularly for keeping the levels of bacteria in the large intestine at the right number. Again, the big concern with having these bacteria in the large intestine is we don't want these bacteria to be able to get up into the small intestine where we have a lot of absorption taking place. Because we don't want those bacteria, even though they're great in the large intestine, we don't want those bacteria to end up into our bloodstream because then we would become septic, and that's not good. So the big problem with appendicitis, when that appendix becomes inflamed, the big problem is it can rupture. And if it ruptures, then you have the risk of bacteria that could either be in the appendix or here in the large intestine. Big concern is that it could end up out here in the abdominal cavity and then go into blood vessels, a lot of which are here in these mesenteries, and we don't want that to get into the bloodstream. So you have to be real careful with appendicitis. You need to treat it right away and make sure that you don't let those bacteria get into the bloodstream and get out of control. Now, for appendicitis, you're most at risk for appendicitis during adolescence. And the reason why is the appendix during adolescence is at its widest. And so you have to be really careful with it at that point. As you get older and the opening to the appendix gets smaller, there's less of a risk that you'll end up with appendicitis. Now, what appendicitis is, again, it's any kind of inflammation of the appendix. And what usually happens is you'll have fecal material that ends up blocking the appendix. And that, again, can trap bacteria in there. That can cause an inflammation of the appendix. And if it becomes too inflamed, then it can rupture. So that's the big concern there. So, let's see. I think we covered all the different parts of the colon. Again, thing to keep in mind is these different parts of the colon are going to be connected by mesentery. And you can see that here. Here's the mesentery that's connecting the different parts of the large intestine or different parts of the colon together. Remember that in these mesentery, which is just a double layer of connective tissue or double layer of this peritoneum that's surrounding the organs. You're going to find a lot of blood vessels in here that are being routed to the various organs. You'll also find a lot of lymphatic tissue in here, and you'll find some nerves running in there as well. Okay, so let's talk a little bit about these valves and sphincters. So we've seen valves throughout the whole digestive tract. And again, coming into the large intestine, we're going to have the ileocecal sphincter or ileocecal valve that is controlling movement from the small intestine into the large intestine. And then you're also going to find two very distinctive sphincters in the anus, and then 3 different valves or sphincters in the rectum. And these are doing different jobs. The ones that are in the rectum, these guys up here, they're helping to keep fecal material in the rectal area while still allowing gas to be able to leave. And the reason that there's gas being produced, and we'll talk more about this later, but there's bacteria in the large intestine. And again, those bacteria are there. We want them to be there. They're beneficial. But when they undergo various chemical reactions, some of their byproducts are going to be acids and gases. And so those gases we need to eliminate from the digestive tract so that they don't cause discomfort and just kind of build up. So we want to be able to eliminate those gases, but we don't necessarily want that fecal material to be leaving yet. Now, in terms of controlling when the fecal material does leave, that's going to be due to these two sphincters here in the anus. And we have two sphincters here. You have what's called the internal anal sphincter, and this is going to be composed of smooth muscle. And then lateral to that, you have the external anal sphincters, and these are composed of skeletal muscle. And normally, both of these sphincters are going to be closed, meaning that these groups of muscles are contracted, and that's going to keep the fecal material in the I guess, anus at this point until you're ready for defecation. And we'll talk about the reflex that triggers defecation. But ultimately, what we want to have happen is when we decide that we want to defecate, then we want both groups of these muscles to relax, causing those sphincters to open, and then the fecal material can leave. Now, obviously, the smooth muscle, the internal sphincter, we don't have control over that. So whether that's contracted or relaxed is going to be driven by reflexes, and we'll look at that here in a second. But we do have control over this skeletal muscle here, the external sphincter. And so that's going to allow us, up to a certain point, to have control over when we do defecate, because we can decide if this group of muscles, if this external anal sphincter is going to relax or not. Okay, so let's look at the different tunics here in the large intestine and talk about any adaptations that we might have here. So again, when you look at, it's going to have all 4 distinct tunics, just like what we've seen in the esophagus and the stomach and the small intestine. When we look at the mucosal tunic, the epithelia is going to be simple columnar epithelia, just like it was in the small intestine, just like it was in the stomach as well. Now, embedded in that epithelia, we're not going to have any cells or glands releasing enzymes because we're not doing any chemical digestion, because we're not absorbing very much, a little bit, but not very much. We're not going to need to increase surface area, so we're not going to have microvilli or anything like that. So in that respect, no real special adaptations of that mucosal tunic. What we will have, we'll have what are called crypts. And we also had crypts in the small intestine as well. And associated with these crypts and then embedded in the epithelia, we'll have goblet cells. And these structures are going to secrete mucus. And we saw goblet cells throughout the whole GI tract. But here in the colon, we're going to see a lot of these goblet cells. And it's one way that I know that I'm looking at the large intestine or at the colon, is I'm going to see no microvilli, no villi, no gastric pits. I will see simple columnar epithelia, and I'm going to see a whole lot of goblet cells embedded in that epithelia. And again, those goblet cells are secreting mucus, and that's going to help with the passage of the feces. So that's how I know that I'm looking at the large intestine or the colon. I'm looking for the absence of things and then the presence of a whole lot of goblet cells. Now, when we switch over to the anal canal, so when we go from the rectum over to the anal canal, then we're going to switch our epithelia. So in the rectum is still going to be simple columnar epithelia. When we go over to the anus, to the anal canal, now we're going to see stratified squamous epithelia. And that's there because we're going to have a lot of abrasion with that fecal material being compacted in this area and then being eliminated from the body. So we need that nice, strong, tough, durable epithelia here. Now, also in the anal region, you'll have what are called sinuses. And these are kind of like grooves in the wall of the anus, and they're going to help release mucus and also help to compact that fecal material. So if we just look at the anal canal, you can see these grooves in here. Those are the anal sinuses. And again, as that fecal material comes down here, it helps to compact it. And then you can also see here the different sphincters. So this right here would be the internal anal sphincter, and then lateral to that would be the external anal sphincter. And again, normally nerves are coming down and they're contracting those muscles so that both of these sphincters are closed, and that keeps any fecal material that's here or in the rectum or maybe even in the sigmoid colon from being able to come out. Okay, so let's talk about these bacteria that are here. There are quite a variety of bacteria in the colon. There are over 700 different species, and I don't remember the exact number, but it's something like, when you look at your fecal material and you weigh it, it's something like 50% of the dry weight of your fecal material is bacteria, like mostly dead bacteria. I might be off with the numbers, but it was a really high percentage. I remember thinking, oh my God, that's really high. So there's a lot of dead bacteria in there, and then there'll also be some live bacteria as well. Now, the bacteria, how do they get there in the first place? So the bacteria are going to get into the colon when we're very young, and they're going to come through the GI tract. So there are going to be some bacteria that are able to survive going through the esophagus, through the stomach, through the small intestine, and actually make it down to the colon. Plus, you're also going to have bacteria coming up through the anus. And I know that doesn't sound very appealing, but it happens, right? It's an opening. And those bacteria are going to get in there, and they're going to start colonizing the colon. And again, we want this to happen because some of the things these bacteria are doing is they are making and releasing vitamins. In particular, they can make B-complex vitamins like B12, and then they also make vitamin K. Now, we're getting these vitamins through the foods that we eat as well, but these bacteria are helping us to get these vitamins. Now remember, the B vitamins are going to be involved in protein synthesis kind of across the board. But in particular, they're going to be important with red blood cell production because there's so many proteins, hemoglobin in particular, in our red blood cells. Vitamin K, if you guys remember, vitamin K is involved in the clotting process. So I think we mentioned this last quarter. Newborn babies, they don't have these bacteria nice and colonized there in the colon. So if a newborn has to undergo surgery, a lot of times, almost exclusively, they'll give that newborn baby a shot of vitamin K to make sure that their levels are adequate in case they have to have surgery, they can form clots, and there's not going to be a problem with bleeding. Over time, as the bacteria come in and colonize, then you wouldn't have to worry about things like that. Now, these bacteria also can digest some types of carbohydrates that we don't have enzymes to digest. So if we just review this. So if we think about carbohydrates, big carbohydrates, or polysaccharides, there are basically 3 different types that we're consuming. So first of all, for animals, when animals store their excess glucose, and this is any animal, whether it's humans or any other kind of animal, when we store our excess glucose, what do we store it as? Glycogen. And glycogen is going to be stored in liver cells and muscle cells. It's the only place that you're going to find glycogen. So glycogen is a storage form of glucose, so that if we're low in glucose, we can tap into that glycogen, release glucose, and then other cells can use it. And so when you eat any kind of animal product, some of the carbohydrates that you're getting from that animal are going to be in the form of glycogen. And we have enzymes that can break down glycogen, and we can get that glucose, and our bodies can use it as a nutrient source. Now, plants also can store their excess glucose so that they can tap into it later to use it as energy. And what do plants store their excess glucose? As starch. So when we eat, whatever, potatoes or any kind of vegetable, a lot of the carbohydrates that we're consuming are starches. So again, this is how plants store their excess glucose as starch, and then if they need that glucose, they have enzymes that can break that starch down, and then they have glucose available and they can use it. Now, we as humans, when we go to eat plants, we can take that starch and we can break it down because we have amylases. And amylases are taking that starch, breaking it down into glucose. We absorb those glucose molecules, and then our bodies can use that glucose, or we can store it as glycogen, or we can turn it into fats. Now, plants also are going to have another type of carbohydrate, and that's cellulose. And what cellulose is, this again is a bunch of glucoses strung together, but this is not a storage form of glucose. Cellulose is, if you look at plant cells, they're going to have a plasma membrane, just like our cells do. But then plant cells are going to have an extra boundary, or an extra barrier, called a cell wall. It's kind of extra protection for that cell. And it also helps the cell from being able to burst or become too dehydrated. And that cell wall is composed of cellulose. So here, cellulose is a structural component of plant cells. They don't take that cellulose, break it down, get glucose, and use it for energy. If they need glucose for energy, they'll break down their starch, just like we will break down our glycogen. Cellulose is a structural component. Now, the problem is when we go to eat plants, again, we have enzymes that can take the starch, break it down, and we get glucose from that. But we don't have enzymes that break down cellulose. So when we eat plants, we can't do anything with this cellulose. We can't tap into the nutrients that are in that cellulose. Instead, we're going to use that cellulose as fiber. So that's basically what fiber is. Fiber is cellulose. We can't break it down and use the glucose from it, but fiber is good for our digestive tract because it makes us feel full. It's bulky. It bulks up the chyme and then the fecal material, and that also helps with motility. If we have a lot of fiber in our food, it causes the GI tract to stretch, particularly down here in the colon. And when those smooth muscles are stretched, they will inherently respond by contracting, and that helps with peristalsis. Now, back to our bacteria. Some of the bacteria in our colon will be able to break down certain types of cellulose. So we don't have enzymes to break it down, but some of the bacteria can break down certain types of cellulose and use it as a nutrient source for themselves. Now, that's all fine and dandy, but the problem is when bacteria use that cellulose, then they start releasing a lot of byproducts of different chemical reactions. and those are acids and various gases. So that's why if you eat a meal that is really high in certain types of cellulose, you can feel very gassy because the bacteria are going to town on that cellulose, and now you're producing a lot of gas. Right? Beans are a classic example. For some people, they're very sensitive to broccoli. This can also cause the same thing. So that's what's going on there. Now, other animals, like cows, they do have the enzyme to break down cellulose. So some animals can get nutrients from cellulose. We as humans can't. We only get nutrients from starch and then from glycogen. And obviously when you eat any kind of animal product, most of what you're getting is going to be proteins. You're only going to get a little bit of carbohydrates from those animals because you're only going to find glycogen in small amounts in liver cells and muscle cells. So if you're not eating liver, then there goes a lot of the glycogen. Most of what we're eating are skeletal muscles, and so you'll get a little bit of glycogen, but not a whole lot. So animals aren't going to be a good source for us for carbohydrates. Okay. So we talked about what's going on in the large intestine. Material will stay in the large intestine around 12 to 24 hours. So we said in the stomach, it's there about 4 to 6 hours. In the large intestine, about 12 to 24 hours. In the small intestine, somewhere in the middle. Now, because the large intestine is reclaiming vitamins, water, and electrolytes that didn't get claimed at the small intestine. Again, most of this is happening in the small intestine. But because we're only absorbing a little bit of these things at the large intestine, we really can get by without having the colon. The main thing that the colon is doing is absorbing these last little bit of things we didn't get in the small intestine and then producing fecal material. So if for some reason you had to have part of your colon removed, you would still be just fine. It would not be that problematic. So if you have to have something removed in the body, colon wouldn't be, would be higher on the list of things to get removed if you had to have something removed. And they can basically, if they had to, they could link the ileum right down to the anus so that you could release material. Obviously, it would tend to be very watery because the reason that you have the semi-solid fecal material is the longer that it stays in the large intestine, the more water is getting reabsorbed. Now, this is also why you want to make sure that you have decent motility. If fecal material is staying in the large intestine more than this, more than 24 hours, then the problem's going to start being that fecal material is going to start to become very hard and very dry because you keep absorbing water from it. And this is how you end up with constipation. If you have material going through the large intestine faster than this, then you're going to end up with diarrhea because you don't have the chance to absorb that water. So you want a nice, good motility so that things aren't too watery and you have the potential of getting dehydrated or too hard. Then you have the potential for being constipated and then you get cranky and it's just no good. So in terms of motility through the GI tract, most of the time the large intestine really is not doing a whole lot. It's relatively inactive, and then you'll have these slow, what are called haustral contractions that mix and then propel the material forward. And these are kind of slow moving contractions. But as you have material in the stomach, that will trigger reflexes that cause the large intestine to become more active. Again, it's the whole thing of put food in the stomach, you want to move previous material in a forward direction so that that food then can eventually get to the small intestine and large intestine. Have material in the small intestine. That will trigger a reflex that causes the large intestine to become more active. Have material in the large intestine. That will cause distension, and that will cause the smooth muscles to start to contract. So you've got several different reflexes. We talked before about the gastroileal reflex. So again, this is when you've got food going into the stomach. That's going to trigger a reflex that causes the smooth muscle of the ileum to start to contract. That's going to move material, chyme, out of the small intestine into the large intestine. You'll also have other reflexes, like the gastrocolic reflex. This would be eat a meal, put food in the stomach. This will then set up a reflex that causes the smooth muscle of the colon to contract. And this will help to move material that's in the colon down into the anus. And this is the major reflex right here, where people say, gosh, I just ate and my GI tract is so active, my metabolism is so high, I go to the bathroom within 30 minutes. So it is not that food that you are defecating, right? A lot of people think you eat and then 30 minutes, I've got great metabolism, I'm defecating that. No, right? When you eat, that triggers a reflex that if you do have material that's in the colon, far enough in the colon, that can cause that material to go down into the anus, and then shortly after you'll have the urge to go to the bathroom. So several different reflexes being set up. Here are two. Again, as you have material in the large intestine, it will stretch. That will activate stretch receptors. That will set up a short reflex, causing the smooth muscle right there in the colon to contract, and that helps to move material forward. So again, you want fiber in there. It helps to activate these reflexes. It helps to move material in a forward direction. Okay, so the defecation reflex. So basically this is a parasympathetic mediated reflex. And again, most of the time, both of those anal sphincters are going to be closed, meaning that you're going to have nerves coming down and stimulating those muscles so that they are contracted. So what's going to happen is when you get material, when you get fecal material that moves down into the sigmoid colon and also down into the rectal area, that's going to activate stretch receptors in the wall of those different areas. And those stretch receptors then are going to send information along visceral sensory neurons up to the spinal cord. And we're going to process that material or that information in the spinal cord. And coming out of the spinal cord are going to be parasympathetic nerves that come down and cause that internal anal sphincter to relax. Now, that's great. We've got half of it done. But we also then have to have that information going past the spinal cord, that sensory information going up to the brain, up to the cerebrum, where we process it and say, oh, I have the urge to go to the bathroom. So that visceral sensory information is going up to which area of the brain where do you think that type of information would go? What cortex do you think? Sensory info.
Oh.
General sensory info. It's visceral, but is that visual cortex?
Is it parietal?
Yeah, you're getting close. What cortex is that?
Here.
Like, so it would be close to the somatosensory cortex. It wouldn't be somatosensory, right? Because that deals with skeletal muscles. but it would be in the parietal area somewhere, around the parietal and frontal lobe areas. Okay, so it's kind of similar to somatosensory, it's just visceral sensory. So that information's going up, you're going to process it up there in the cerebrum, and then we're going to activate the motor cortex in which lobe? The frontal lobe, okay, on which gyrus? Precentral gyrus, okay? And coming down from the precentral gyrus, the motor cortex and the frontal lobe are going to be somatic motor neurons that then come down to the external anal sphincter, cause it to relax, basically inhibiting those pathways. And both sphincters then will be open and then defecation occurs. However, if you decide that you don't want to respond to that stimuli right away, then you can ignore it, right? You can ignore that and you can keep those action potentials coming down and keep the action potentials firing these skeletal muscles of the external anal sphincter, and you'll just have one of the sphincters open, one closed, and you won't go to the bathroom. And, you know, depending on how strong the stimuli is, it might be 30 minutes later, it might be an hour later, this reflex will get set up again. Usually once you have a little bit of movement happening, it'll trigger that reflex again. And again, it will cause the internal anal sphincter to open, the external anal sphincter, you'll be aware of it, and then you can decide whether you want it to relax and open or not. Now you can do that up to a point, but once you get a certain amount of fecal material in either the sigmoid colon or the rectum, and if those stretch receptors become so active, then you're going to lose the ability to control that reflex. And both of the sphincters are going to open due to so much pressure being generated in here. So if there's a certain amount of pressure because of the quantity of material down here, then that can override these external anal sphincters and force them to open, and then defecation happens. When you get older, it starts to become more difficult to keep these sphincters contracted. So as you get older and you start to lose the number of neurons that you have, then that means you're going to start to lose control over muscles. So the less neurons you have, the less able you are to keep those muscles contracted, and the more likely it's going to be for you to have accidents. So that's what we have to look forward to as we get older. Now, these last several slides, I'm really not going to say much about vitamin absorption, mineral absorption, or water absorption. What I want you to know is that most of those compounds are getting absorbed in the small intestine. The large intestine is where we can finish up absorption of those particular compounds. But again, remember, monosaccharides, fatty acid glycerols, and amino acids, that's all happening in the small intestine, not the large intestine. And that has to do with what kind of transporters you find on the epithelial cells. Now, one thing to mention about vitamins is, again, we're going to take these different vitamins in through our diets. The B vitamins and K vitamin we can also make via these enteric bacteria. Now, we have, in terms of vitamins, you can break them down into, are they fat soluble or are they water soluble? And this will influence how they're absorbed and how they're stored in the body. So your fat soluble vitamins are A, D, E, and K. And these guys are going to get absorbed with fats. So it's very important, if you want to be getting these vitamins into your body, you have to eat a certain amount of fats. It doesn't need to be very much, but you have to have fats present for those vitamins to get absorbed. Otherwise, they'll just go right through the GI tract. So they're going to get absorbed with your fats through the lacteals and then eventually end up in the circulation. Now, the water-soluble vitamins, these would be your B's and C's, and these are going to get absorbed via little pumps or little transporters. Now, remember that the exception here is vitamin B12. Vitamin B12 will only get transported if intrinsic factor is also present. So intrinsic factor has to be present. It's made by the parietal cells of the gastric glands in the stomach, and that's how you're going to absorb that. Now, once you get these various vitamins into your bloodstream, if they're water-soluble vitamins, your body's going to use those particular vitamins as needed, and then whatever it can't use, then those vitamins are going to just end up in your urine and being excreted from the body. On the flip side, your fat-soluble vitamins, they will get absorbed in your adipose tissue. So these can build up and be stored in your body. So if you're taking mega doses of vitamins, you have to be very careful because these can start to become toxic at certain levels. So you should always be careful about how much vitamins you're consuming. And if you are consuming vitamins as a supplement outside of just eating a balanced, modified diet or varied diet, you have to be careful. You should always take food with your supplement, and you should always make sure that that food has a little bit of fat in it so that you can absorb these guys. Otherwise, you're just going to be, your urine's going to start to become more costly, okay? Because most of it's just going to end up in the urine. Now, yeah.
So when we are releasing the fat-soluble vitamins from our adipose tissue, are they released as needed, or is it just kind of a random release of vitamins as we burn off the tissue?
I think, and I'm not 100% sure on this, I think that they are released as you break down adipose tissue. I don't think that there is like some homeostatic mechanism that kicks in and says, if you're low in A, that will cause A to be released from your adipose tissue. But I'm not 100% sure on that. But I've never seen anything talking about homeostatic mechanisms of these vitamins here. Yeah.
So taking excessive amount of fat-soluble vitamins could lead to gaining fat?
No, it won't cause you to become fat because these essentially are, they're not, so if you're eating a lot of extra calories, right, those calories will get converted to fat and then you'll store them. Mm-hmm. These guys are just minuscule. So if you're consuming a lot of these, it's not going to add really to your overall weight like that. It's not going to be, they're not bulky like storing fat is. They're just stored in the fat, but they're not going to be like bulk like the adipose tissue is. But the big thing is you have to worry about toxicity levels. They can become toxic. Okay. For your electrolytes, again, just remember that most of these are getting absorbed in the small intestine. And remember that the way that these various ions or minerals or electrolytes are going to get absorbed is by some kind of protein transporter or protein channel. That's how they're getting absorbed. And we're not going to talk about different gradients and whatnot. Don't worry about that. One thing to keep in mind, remember that with calcium absorption, we have to have vitamin D present. So if we don't have vitamin D present, then those calcium transporters are not going to work as efficiently. So this is how, we've mentioned this several times, PTH is going to influence calcium absorption because PTH will go to the kidneys, cause more vitamin D to be produced. Vitamin D then will go through the bloodstream to the small intestine, to the GI tract, and cause increase in calcium absorption. Water, same thing. Water is basically going to get absorbed via osmosis. So the more sodium that we absorb, the more water that we will absorb as well. And again, I'm not going to ask you details about gradients and transporters or anything like that. Okay. These last slide is just a summary slide of what we've talked about already. This is a nice little summary slide that just is telling you kind of what's going on in the different parts of the GI tract. It's telling you what enzymes are involved, where they're released, where they're active, what they're breaking down. So again, this is a nice summary slide, probably more information here than we went into. But again, when you're studying, you definitely want to make sure that you understand the 4 tunics, and in every organ, what adaptations of those tunics are happening to allow particular function to occur. You want to know what enzymes are released, where they're released, where they're active, what those enzymes are breaking down. So I think if you think about the digestive system, think about structure dictating function, the different enzymes, what they break down, and then homeostatic mechanisms, what causes smooth muscle to contract in a particular area, what causes the pancreas to release its secretions. Those would be the 3 avenues of studying. So the different tunics, structure dictating function, enzymes, mechanisms, homeostatic mechanisms. Yeah.
With people who are lactose intolerant, are they just missing lactase?
That's exactly right. Or it's either they're missing it completely or it's not very efficient. Could be that they just have less of it than somebody else, they're not making as much, or it's just not as effective. So yeah, that's it. So they can't break down lactose, which is the sugar that is in milk products. And so what happens is lactose, then instead of getting broken down, it just continues through the GI tract, and that can be really irritating because the bacteria in there will use it as nutrients, and then they start releasing lots of gases and acids, and you get real bloated and a lot of discomfort. So that's what's happening. So pretty much all of those sugars that we take in are getting absorbed in this by the small intestine. Lactose gets to keep going through the GI tract, and the bacteria just have a field day with it. So, switching gears over to the urinary system. So we're going to approach the urinary system like everything else that we've done. We're going to start out looking at the major structures. And for the urinary system, it's going to be pretty straightforward with function or structures. And then we're going to spend most of our time talking about the nephron, which is the smallest functional unit of the kidney, like the sarcomere was in the skeletal muscle, like osteons are in compact bone. And we're going to look at what happens at this nephron. And we're going to spend a lot of the time talking about how the urinary system regulates blood volume, blood pressure, how we can increase or decrease how much sodium we're reabsorbing, how much calcium we're reabsorbing. So we're going to look at a lot of homeostatic mechanisms in the urinary system. And we're going to talk a lot about hormones in the urinary system, because the nervous system's not going to be a big player here, but hormones will be. So when you look at the urinary system, 3 major things that the urinary system is doing. First is excretion. We've got to get these organic waste products that our cells are producing through various chemical reactions. We have to get them out of the body, out of the bodily fluids, which would be extracellular fluid, blood being an extracellular fluid, because those products are toxic to the body, toxic to the cells, so we have to get rid of them. We have to eliminate those products, so we've got to get them out of our bodily fluids, out of our blood, and into the urine, and then we've got to get that urine out of the body. Okay? And then we're going to do homeostatic regulation, primarily of blood volume and thus blood pressure. And then also we're going to tweak the solute concentration of our blood. Make sure that we have the right amount of sodium in there, the right amount of calcium, the right amount of everything in our blood. So that's the solute concentration of our blood plasma. So when we look at structures, again, pretty basic structures. Obviously we've got 2 kidneys, and they're going to be located dorsally in our abdominal cavity. Coming out of each kidney on the medial side will be tubes called the ureters. The ureters are going to carry the urine that's produced by the kidneys down to the bladder, and the bladder is going to store that urine. And that's the only thing the bladder is doing, is storing that urine. And once we decide that we want to release or eliminate, then the urine is going to travel through the urethra and out the body. So you can see on the medial side of the kidneys here, just like when we looked at the lungs, on the medial side of each lung, we're going to have things entering and leaving the kidneys. And those things would be the renal artery will come in and branch. The renal vein is taking blood away from the kidneys, and then the ureter is carrying the urine out of the kidneys. And that general area there on the medial side of the kidneys, where we've got things entering and leaving, that's called the hilum or the hilus, just like it was in the lungs. So don't go trying to find a particular spot that is the hilum or the hilum. There is none. It's a general area on the medial side of the organ where things enter and leave. That's the hilum. You can see on top of each kidney is the adrenal gland, but what goes on in the adrenal gland is completely separate from the kidneys. They just happen to sit on top of there. Now, when we cross-section a kidney and look inside of it, we can see that there are things going on here inside of the kidneys. So what we're going to do is we're going to talk about how blood flows in and out of the kidneys. We'll do that here in a minute. But we're going to take that blood that comes in, and we're essentially going to filter that blood. And we're going to take a lot of stuff out of the blood. And then as that fluid that we took out of the blood is traveling through the kidneys, that fluid will be called the filtrate. As it's traveling through the kidneys, we're going to be able to reabsorb material, meaning take material that ended up in that filtrate, and we're going to be able to put it back into the blood. And then we're also going to be able to secrete. We're going to be able to take things that are in the blood and put it into the filtrate. So we'll talk in a lot of detail about this. So as that blood comes into the kidneys, we're going to filter, we're going to reabsorb, and we're going to be able to secrete. Those are the different processes that are going to happen. And once we're done with those processes, we're going to end up with a fluid that will then be the urine. Now, as the urine is produced, it's going to, and we're going to look in detail about how that happens in here, but the urine is going to leave through what are called the renal papillae or papillary ducts, and that they're at the base here of each of these renal pyramids. So these guys are the renal pyramids. At the base of those renal pyramids are papillary ducts or renal papillae, same thing. The urine's going to go out of those renal papillae and then it's going to collect in these little grooves right here called the minor calyces or minor calyx. And then the urine from the minor calyx is going to drain into a major calyx, and then urine from the major calyces is going to drain into the renal pelvis, and from the renal pelvis it's going to go into the ureter. So urine's going to be produced. Again, we'll talk about that happens. It's going to go through the base of all of these renal pyramids through a duct called the renal papillae or the papillary duct, then into the minor calyces, major calyces, renal pelvis, ureter, down to the bladder. Now, when you look at the kidney like this, the outermost part of the kidney is called the cortex, and then everything kind of towards the middle is called the medulla. Now you can see that in the medulla here, there's lots of things going on. We've got these renal pyramids, we've got the calyces, we've got the pelvis, renal pelvis. All of that's part of the medulla here, this middle part. The cortex is just this outermost edge of the kidney. Now, what we're going to see is millions of really small units are kind of snaking their way through the cortex, down through the medulla, back up into the cortex. These are going to be the nephrons. And again, they're going to be little tubes that are coming from the cortex down into the medulla in these renal pyramids and then back up into the cortex. And we're going to look at those in a lot of detail. But every kidney is going to have, about 1.5 million or so nephrons. And again, they'll be all here in the cortex and then in the renal pyramids in the medulla part. Now, these nephrons, they're going to be a series of tubes, essentially. And if you would pull them all out and put them end to end, there would be about 85 miles of tubes of these nephrons in each kidney. So just think about, that's a lot of tiny little tubes that are all within the kidney. Very similar to when we talked about the seminiferous tubules in the testes. A lot of really tightly coiled tubes here in the kidney. Now, when we look at the nephron has different parts to it. And when you look at the nephron, you're going to have what's called the renal tubule, and then the renal corpuscle and the renal tubule. And the first part's going to be the corpuscle, and then we're going to go into the tubule. Just draw this out here. Okay, so first part is the renal corpuscle. So this is all part of the nephron. And again, you're going to have about 1.5 million of these guys in each kidney. So the nephron consists of two major parts, the renal corpuscle and then the renal tubule. Now, the renal corpuscle has different parts to it. The renal corpuscle consists of this hollow ball here, which is called Bowman's capsule. Or you'll also see it referred to as the glomerular capsule. Same thing. And again, this is like a hollow ball. And if you look inside of that ball, there's going to be a mass of capillaries in here. And those capillaries have a special name. They're called the glomerulus. But they're just capillaries, like capillaries we talked about before, they're going to have some special features to them that we'll discuss. So the renal corpuscle is the first part of the nephron. It consists of Bowman's capsule, and inside of that capsule is space called the capsule space or Bowman's space, and the glomerulus, this massive capillaries. Then the second part of the nephron is called the renal tubule, which is just a tube that this fluid is going to travel through. And there are different parts of the renal tubule. You have the first part that comes off of the corpuscle here, comes off of Bowman's capsule. The first part is called the PCT, proximal convoluted tubule. Proximal because it's closest to Bowman's capsule, PCT. Then we go into this loop called the loop of Henle. And you'll also see it called the, what else do you see it called? The renal loop. Renal loop, I guess. The loop of Henle. And the loop of Henle has a descending limb and then an ascending limb to it. And we'll talk in more detail about that loop later. And then we go into the DCT, the distal convoluted tubule. And then we're going to have different DCTs from different nephrons. So I'll just put one over here. They're all going to converge and drain into a collecting duct. So one more time. The nephron consists of two major parts, the renal corpuscle and the renal tubule. The corpuscle consists of a hollow ball, Bowman's capsule. Inside of that ball is space and a big clump of capillaries called the glomerulus. The tubule is a tube, and it consists of different parts. You've got the PCT, proximal convoluted tubule, and then the loop of Henle, and then the DCT, the distal convoluted tubule. And DCTs are going to drain into the collecting ducts. So what you're going to have is, you're going to have this filtrate being produced. We're going to filter the blood, and that's going to happen here between the glomerulus and Bowman's capsule. And again, we're going to go in lots of detail about what's going on there. But this is where we're first going to filter the blood, is at this massive capillaries. And we're going to produce a filtrate, a fluid, that's mostly water, but with a bunch of stuff in that water. Okay? That filtrate then is going to leave the corpuscle, and that filtrate is going to start traveling through the tubule. So that filtrate's going to go through the PCT, the loop of Henle, the DCT, and then that filtrate is going to drain into the collecting ducts. And then we're going to go, that filtrate, once it gets into the collecting ducts, then it's going to go through the renal papillae, and then into the minor calyces, and then into the major calyces, and then it's going to drain into the renal pelvis, and then it's going to leave the kidney through the ureter. Down to the bladder, and then it's going to leave the body via the urethra. So once that filtrate leaves the collecting duct, then it is officially urine at that point. So once that filtrate leaves the collecting duct, we can't make changes to it anymore. So all along this renal tubule, we're going to be able to change that filtrate. We're going to be able to put maybe more things into it, maybe take some things back. But once that filtrate leaves the collecting duct, we can't change it anymore, and it now is urine. And that urine's going to go through the renal papillae, into the minor calyces, drain into the major calyces, into the renal pelvis, ureter, bladder, urethra. And all along this way, you've got more and more urine converging into each other until finally it all accumulates down here in the bladder. Now, questions on that? Don't get behind with anything because you'll get confused. So what we want to do now is we want to say, how does blood flow in and out of the kidneys? Because that's what we ultimately have to do, is we have to filter the blood to make this filtrate. Okay? So I'm not going to draw it exactly the way that, I'm not going to draw the vessels exactly the way they are, because this diagram will get really messy really fast. Okay? But so blood flow in and out of the kidneys. Here's kind of a schematic of it, and this is more detailed than what I'm going to ask you guys. But actually, before we do that, let's put this into context right here. What we're looking at here, okay, is if we look at the kidney, so we can see a nephron right here. The nephron's going to start up in the cortex of the kidney, and then it's going to dip down into these renal pyramids, into the medulla, and then it's going to come back up into the cortex, and then it's going to dip back down into the renal pyramid or the medulla. And then the renal papillae are here at the tips, and then the calyces, renal pelvis, ureter. So when we're looking at this right here, this top part, so I'd say about like that. This right here is the cortex, and going this way is the medulla. So if we take a little chunk out here, if we take one of these renal pyramids and pull it out here, then up here in the cortex, that's where we have the renal corpuscle, and then most of the PCT is going to be up there in the cortex, and then the loop of Henle is going to dip down like this into the medulla, and then the DCT is back up in the cortex, and then the collecting duct is down here into the medulla, and down here at the tip is the renal papillae, and then you go into the minor calyx, and then major calyx, renal pelvis, ureter, down to the bladder. Okay? So that's how it's dipping through the cross-section of the kidney. Now, blood flow in and out of kidneys. So coming into the kidney, we've got the renal artery coming into the kidney. The renal artery is going to branch once it gets into the kidney, and those arteries are then going to branch into smaller and more numerous arteries. Okay, and we're not going to talk about all those different arteries, but blood flow. So first of all, we've got the abdominal aorta. So we've got, might as well put the heart up there. So coming off of the left ventricle, we've got the aorta. And we go down to the abdominal aorta. And what are the branches half of the abdominal aorta is the renal artery. And that's bringing blood into the kidney. Now, we're going to have, like I said, several branches once we get into the kidney that we're not going to talk about. Okay? So you can see there's several different arteries. And eventually, we're going to get down to an arterial. Just like every time we've talked about circulation, that's been the pattern that we've taken. Now, this arterial has a particular name, and it's called the afferent arterial. And arterials take us into what? Capillary beds. And the first capillary bed that we're going to encounter is called the what? The glomerulus. Now, normally, leaving a capillary bed, we would go into what? We would go into a venule next. But we're not going to do that here in the kidneys. We've got a different schematic that's happening. So, leaving this glomerulus, instead of going into a venule, we're actually going to go into what's called the efferent arterial. And that's going to take us into a second capillary bed. And these are called the peritubular capillaries. And leaving the peritubular capillaries, now we're going to go into a venule, and then those venules are going to drain into veins, and those veins are going to converge into larger and less numerous veins until eventually we are leaving the kidney through the renal vein. Leaving the kidney through the renal vein, and the renal vein is going to then drain into the what? Inferior vena cava. Great, and eventually that's going to get us back to the heart. So, again, you can see it's all written right here. We're just not learning all the different arteries and veins, small ones, that are in the kidney. We're just focusing on the main artery and vein that takes us in and out of the kidneys. And then we're concerned with the capillaries. Because the capillaries is where we're going to have things diffusing in and out of the blood. Remember, we can't have things diffusing in and out of the blood in arteries or arterioles. Why? Why can't we? They're too thick, right? We can only have things like water and glucose and organic wastes and other things moving in and out of the blood at capillaries because they're a single cell layer thick. They're simple squamous epithelia, and that's it in the wall. So, if we take this blood flow here and now put it in reference to our nephron, we've already put our glomerulus up here. So we said our glomerulus is in Bowman's capsule. And we said bringing blood into the glomerulus comes in through the afferent arterial. So we better put an afferent arterial in here. So this would be our afferent arterial. And blood's going to leave the glomerulus through an efferent arteriole. So I'm just going to put the efferent arteriole right here. And then from the efferent arteriole, we go into the peritubular capillaries. So if you look at blood, it's coming into each nephron through an afferent arteriole, and then blood's going to go into a glomerulus, and then it's going to leave the glomerulus through the efferent arterioles, and then that blood's going to go through peritubular capillaries. And this is happening at every single 1.5 million nephrons. And the peritubular capillaries, they are all around this renal tubule. So all around this renal tubule are the peritubular capillaries. They're all around here. Yeah, that's.
Do they have two different jobs, the glomerulus and the peritubular, or is it just two times that it's filtered?
No, they're going to do different things. And we're going to get there. The main thing at this point is to know that at the capillaries is when things can move across, in and out of the blood. And we're going to talk about whether things are moving in or out and what that's called in just a second. So at every single nephron, we're going to have an afferent arteriole bringing blood into the glomerulus, an efferent arteriole taking blood away from the glomerulus and into the next capillary bed. And that capillary bed, instead of being at just one little spot, just so happens that those capillaries are wrapped all around this tubule. Now it looks big here, but remember there's 1.5 million of these here in the kidneys. So it's really not taking up very much space. Those capillary beds are they just happen to be wrapped all around that tubule. And then blood that would be leaving these capillaries, these peritubular capillaries, that blood would then flow into venules. So you're going to have venules from different nephrons collecting blood. And then eventually all that blood will go into the renal vein and then out it goes. And as that blood is leaving the kidneys, filtered and clean, urine is traveling through the collecting duct to renal papillae, minor calyx, major calyx, renal pelvis, ureter, bladder, urethra, out in the body. So blood comes into this nephron, and from that blood we are going to make a filtrate. We are going to make urine. And then the clean blood goes back into the system, and all the crap that we don't want goes down to the bladder to eliminate. Okay.
So you can blood in your urine. What's that?
You can have blood in the urine. You don't want that to happen. So that's when things are not working properly, because blood cells should be too big to cross these capillaries, but they do in that case.
That's where it's doing that. That's where it's happening. Blood is mixing.
Yeah, right there. Okay. Questions on this? A few more things before we stop. Okay, so now we want to say, how do we make this, how do we get this filtrate? How do we get this urine? And again, there's going to be 3 processes that we're going to do. We're going to do filtration, we're going to do reabsorption, and we're going to do secretion. By the time we're done with these 3 processes, we're going to have urine that's going down to the bladder, and blood, clean blood, that's going back into the general system, systemic system. Okay. Now again, remember, in the corpuscle is where we have the glomerulus, Bowman's capsule. And this is the first time that we're going to encounter blood that we can do something with, because it's the first time that we hit a capillary bed. And what we're going to do here is called filtration. So what's going to happen here in the corpuscle, we're going to see, and we're going to talk about why this is the case, but we're going to see that the glomerulus, these are like normal capillaries, except they are particularly leaky. They're kind of a little bit bigger, a little bit leakier, and there's going to be very high pressure in these capillaries, higher than normal, higher than a normal capillary. And what that's going to do is anything that's inside of the glomerulus, if it is, because there's high pressure in the glomerulus, anything that is small enough to fit through these little slits that line the wall of the glomerulus, anything that's small enough to fit through these little slits is going to leave the blood and go out here into Bowman's capsule. So Bowman's capsule is basically a collecting area for this fluid. So because the pressure is high in the glomerulus, so just think, if this right here were Bowman's capsule and I was a glucose molecule inside of the glomerulus, the pressure in the glomerulus is really high and it's forcing me out of the glomerulus and into Bowman's capsule. And anything else that's small enough to fit through those slits. So water, glucose, amino acids, sodium, potassium, calcium. Anything that's little is going to leave the glomerulus and go into Bowman's capsule. And that is the filtrate that we produce. And that process is called filtration. So most of it's going to be water, and then whatever's dissolved in that water that's small enough to fit through the slits, that ends up in the filtrate. Now, the big problem with that is, if you've got blood there in the glomerulus, most of what's in the blood is going to be small enough to fit through those slits, right? Really, the only thing that's not going to be small enough are going to be cells, red blood cells, white blood cells, and then plasma proteins. Those are going to be the things that can't leave the blood, and everything else is going to leave. All that water, all the ions, all the glucose, all the amino acids, plus all the waste and drug metabolites and whatever else is there. So we're going to be producing a lot of this filtrate. And if that filtrate then would travel through the renal tubule, through the papillae, minor calyx, major calyx, renal pelvis, ureter, bladder, urethra, we would be really dehydrated because we would lose most of the part of our blood. We would lose most of our blood plasma. So that means that while that filtrate, as it leaves the corpuscle, as it leaves Bowman's capsule, it starts traveling through the renal tubule. So what we need to do is, most of what we just put in that filtrate, we have to reabsorb it. We have to put it right back into the blood. And that's what reabsorption is. Reabsorption is taking material, water, glucose, amino acids, ions, that's traveling through this tubule that's in the filtrate, and it has to be reabsorbed back into the blood, back into these peritubular capillaries. And what we want to keep in that filtrate would be water and then waste products, things that we don't want in our blood. So filtration happens up here, and that's material moving out of the blood, out of the glomerulus, and into Bowman's capsule. That's filtration. That's where we first produce the filtrate. Then the filtrate starts traveling through these tubes, through the renal tubule, and some of what's in that filtrate is going to get reabsorbed, put back into the blood, back into these peritubular capillaries.
How does it know what to reabsorb?
We'll talk about that. It'll depend on what pumps are there, what gradients, right? Because things are only going to move via diffusion or active transport.
Okay.
So it'll be gradients that are set up in this extracellular space here, and various active transport pumps. And then also, while that filtrate is traveling through the tubule, we can also do something called secretion. Secretion is taking material that's in the blood and putting it in the filtrate. Now, why do we do secretion if we did filtration here? Because it's movement in the same direction. Filtration's going from the blood and making a filtrate. Reabsorption's going the opposite direction, from the filtrate back to the blood. Secretion, same movement as this, going from the blood, putting it in the filtrate. Secretion happens because even though a lot of material is going to get filtered here, not everything is going to get filtered. So maybe we didn't get all the organic waste out of the blood here. While that filtrate is moving through the tubules, we can do secretion. We can have pumps that are taking those organic wastes that are in the blood, and we can pump that material into the filtrate. So that by the time that material, that filtrate leaves the collecting duct, now we can't make any changes. We can't do any reabsorption. We can't do any secretion. Now it's urine, and now it's just going to travel through these tubes and exit the body. And hopefully, by the time that we're done here at the collecting duct, we have just the right amount of water in that urine, the right amount of sodium, the right amount of calcium. We've reabsorbed all the good stuff back into the blood, the glucose, the amino acids, the sodium that we needed, the calcium we needed, the potassium that we needed, so that again, what's in that urine is the stuff that we really don't need. Water, organic waste, maybe some hydrogen ions that we don't need, maybe some sodium that we don't need, maybe some calcium we don't need, whatever in the body. Questions on that?
I have one question.
Mhm.
So I just want to make sure that I understand. So some blood plasma goes into a PCT and some blood plasma goes to appearance arterial.
So think about what blood plasma is first of all, right? So blood plasma is the water and anything that's dissolved in the water. So what ends up in the filtrate is anything that's in that plasma that's small enough to fit through the slits. So that should be most of what the plasma is. The water and anything that's dissolved in it that's small. What shouldn't end up in the filtrate are the big plasma proteins. They should. They're part of the plasma, but they shouldn't end up in the filtrate. So yes, it's true. The filtrate will end up right here looking very much like blood plasma. Most of it will be in the filtrate. Some of it will still be in the blood, but most of it will be in the filtrate. And then as that filtrate moves through the tubule, we are going to then reabsorb most of that right back in the blood to keep our blood volume where it should be. Okay. Now, for some reason, I always get really disconcerting blank stares at this point. So I just want to make sure that you guys have an understanding of what's happening. Okay. Yeah. They should not cross, and we're going to talk about what happens in filtration, we're going to look at that in more detail, but they should be too big to fit through the slits. They're too big. What do pregnant women do? They check protein in their urine? Probably. So what I'm guessing is, so when women get pregnant, their blood volume can increase 50%, which causes their blood pressure potentially to really skyrocket. And so what can happen, because their pressure is so high, they can be having a really high pressure here in the glomerulus, and that could cause things like proteins, plasma proteins, to end up in that filtrate that shouldn't. Yeah, so that's... So that would be why. So again, don't get ahead of yourself. We're going to talk about each of these processes. I just want to make sure you understand the general concept of what's happening at this point. All right. So let's do a few more things before we stop. So when we look at these nephrons, okay, again, this is a nice image of a nephron right here. So this would be the corpuscle. This is Bowman's capsule with the glomerulus inside of it. Afferent arterial bringing blood in, efferent arterial taking blood out. What they don't show you is the peritubular capillaries. And they often don't show you those because the image gets real messy when they do. But coming off of this efferent arterial, then we would go into the peritubular capillaries that would be wrapped all around this tubule. So that's the blood that's flowing. Okay? So you want to be thinking about how is the blood flowing, and then where is this filtrate being produced, and then where does the filtrate flow? Because you've got these two fluids either running through blood vessels or running through the renal tubule. And then things that are in that fluid are going to be crossing from one place to the other. And again, it's all based on diffusion. Things are either going to be diffusing out of the tubule, so across the wall of the tubule, across the epithelia of the tubule, into the extracellular space, and then diffusing into the capillaries that would be right here, again, across the layer of epithelia, simple squamous epithelia. Or material is going to be diffusing across the wall of the peritubular capillaries, into the extracellular space, and then diffusing into the tubule. And again, diffusion can happen pretty easily because the capillaries are a single layer of simple squamous epithelia. The tubule is just a single layer of simple cuboidal, simple columnar-ish type of epithelia. So things are going to diffuse pretty easily in and out. And again, you've got to have gradients. You've got to have something high somewhere and that thing be low somewhere else, so it will diffuse. Or you have to have pumps. And what we're going to see is, throughout this tubule, some things are just going to passively diffuse down a gradient. Some things are going to have to be actively transported. And if they're actively transported, then that means, let's say, in the wall here, you have to have a sodium transporter, and that can transport sodium. Or you have to have a chloride transporter, and that will transport chloride. If you don't have a specific active transport pump, then you're relying on basic facilitated diffusion. Okay? But that's how things are going to be moving. Okay? So when you look at the tubule, again, the filtrate gets produced here. That's not due to any pumps. That's just pressure forcing the material through a little slit. There's no channels, there's no pumps, there's nothing like that happening here. It's just brute force, pressure, and then forcing things through little slits. Then that filtrate is going to travel through the PCT, the loop of Henle, the DCT, and then go into the collecting ducts, renal papillae, minor calyx, major calyx, renal pelvis, ureter, bladder, urethra. Okay. Now, again, throughout here, we can have reabsorption, taking stuff that's in that filtrate, putting it into the peritubular capillaries, or we can have secretion, taking things that are in the blood and then putting them into the renal tubule, into that filtrate. But secretion and reabsorption are going to be due to diffusion or active transport. We've got to have channels, we've got to have pumps, or we have to have gradients. Here, brute force, forcing material through a slit. Now, there are actually two different types of these nephrons, and they're pretty similar. One is called a cortical nephron, and the other is called a juxtamedullary nephron. And the main difference between the two is how long is this loop of Henle? If it's a short loop of Henle, then it's a cortical nephron. And when you look at humans, 85% of our nephrons are cortical nephrons. The loop of Henle, or here it's called a nephron loop, that's the other way to refer to it. Nephron loop, or the loop of Henle, is very short. So that means most of the nephron, if this guy's really short, most of the nephron is up here in the cortex, in the cortical region of the kidney. Juxtamedullary nephrons have a very long loop of Henle. And for humans, again, it's about 15% of our nephrons have long loops of Henle's, which extend really deep into the medulla. Now, the reason this is important is we're going to see that in that loop of Henle is where we're going to reabsorb a lot of water. It's not going to be the only place, but it's a major way to reabsorb water as that filtrate moves through the loop of Henle. And if we only had really short loops of Henle's, we wouldn't be able to reabsorb as much water because the loop is short. There's less surface area for that reabsorption to happen. And we're also going to see that a long loop is going to be important for reabsorbing water because of, as we go deeper in the cortex, the osmotic gradient becomes greater as we go deeper. And that's going to allow for better and more osmosis, movement of water. So for humans, if we only had cortical nephrons with short loops of Henle, we wouldn't reabsorb as much water and we would become very, very dehydrated. Having these 15% long loops of Henle, juxtamedullary nephrons, allows us to reabsorb enough water so that we don't end up really dehydrated. And we're going to see that there are lots of things that will play a role to keep us from being dehydrated. The fact that we have 15% juxtamedullary nephrons helps us from being dehydrated. The fact that we can release ADH and aldosterone helps to keep us from being dehydrated. So there's lots of things that play a role. If you look at camels obviously have to deal with dehydration because of the environment they live in. So camels will have a much larger number of juxtamedullary nephrons so that they can reabsorb more water and store it to keep from being dehydrated. Thanks. Okay. Now, when you look at these juxtamedullary nephrons, they make things complicated because they want to change the name of the peritubular capillaries into something else. And I'm not even going to go there. It's stupid. I don't know why they do that. So we're just always going to call the capillaries that wrap around the tubule, regardless of whether they're cortical nephrons or juxtamedullary. We're just always going to call them peritubular capillaries. I'm not going to change the name because I just think it's silly. No reason for it. So again, these juxtamedullary nephrons allow us to reabsorb more water, and that allows the kidneys to make a nice concentrated urine, and that's what we want. We do not want our urine to look like our blood plasma. We don't want our urine to look like what the filtrate looks like here, because if it did, we would be urinating gallons and gallons of water every day, and we would be so dehydrated. So as that filtrate moves through the tubule, we want to reabsorb most of that water so that our urine is nice and concentrated, and that keeps us from being dehydrated. Now, don't get bogged down by this statement, make a concentrated urine. No, do you want to, when you urinate, you don't want to see your urine bright yellow, okay, or real intense yellow. That's too concentrated. You want it to be very clear looking, but that is still, if you compare that to blood plasma, that is still a very concentrated urine. So don't get bogged down by layman's terms of what concentrated means versus physiologically what concentrated means. Okay, so that's a good place to stop. On Thursday, we're going to take each of these processes and we're going to talk about them in detail and say what causes filtration, what causes reabsorption, what causes secretion, what are the processes and forces behind it, and then we'll talk about how hormones affect what gets reabsorbed or secreted.